I would like to welcome everyone to the IEST webinar P797. Our organization is the Secretariat of ISO TC209 and is an ANSI accredited standards writing organization. Our presenter today is Gordon Ely. He has worked in the pharmaceutical, medical device, and biologics industries for more than 19 years. Gordon is a certified specialist microbiologist in consumer and industrial microbiology and is a registered microbiologist. He is a past president of IEST, the chair of IEST working groups CC20 and CC25. He received his Bachelor's of Science degree in microbiology with a chemistry minor from Brigham Young University. Here is Gordon Ely.
Hello. Welcome to today's presentation. I am excited to share this information with you. I should maybe give Heather a corrected bio there. I'm now up to 24 years in industry, so that's from 5 years ago. But I have also been involved with many writing committees for standards across ISO, USP, AMI, and several other organizations. So I'm excited to share what I know from the USP. Just as background information, the USP is a scientific nonprofit organization that sets standards for food, medical device, dietary supplements. Its mission is to improve global health through public standards, and it's a volunteer organization made up of people like you and I that are able to get on the committees and share our expertise and help build these standards. The USP was founded in 1820, which was something I found interesting because being a microbiologist, microbiology really didn't take effect until the 1840s, 1850s. So now we're at 1820, the USP was already started. If you want to find out more information about USP, you can always log on to their website. Now there's a process that they go through to develop standards at USP. Usually someone will suggest a standard that might be needed to help in the industry, specifically around food and pharmaceuticals. Once USP receives a request like that, or they may have something in place already and they'll revise it, then it will go before an expert committee. There's a high-level group that oversees the writing of standards, and I won't go through this entire process, but basically there's the Pharmacopeal Forum through which proposals are made, and allows public comment period, and the expert committee determines what needs to be done, and with those comments, and they move it forward, or they and vote on it to become a standard. And sometimes most of the time those are approved because they've gone through the process, but sometimes they aren't and they're canceled or reballoted. And if you've been involved in the standard writing process, it's very, very similar. Now the expert committees are responsible to develop and revise the USP standards as they are, and there's actually 7 compendia that make up the Pharmacopeal. There's the USP, which is the United States Pharmacopoeia. There's the NF, or National Formulary. There's a Medicines Compendium, which deals with all the different medications, pharmaceuticals that are out there. There's a USP on compounding, and that's related to our subject today. There's also a compendium on herbal medicines, dietary supplements, and then, if you've heard of it, there's the Food Chemicals Codex. And now there's various committees that help write these standards and revise the standards, and just a few of them a few examples are microbiology, compounding, packaging, biological analysis, and physical analysis. And there's several others. And they make up the groups that write these standards and revise the standards. And you have to be appointed to these committees on it and to be able to give your input. Occasionally, they create expert panels. And the expert panels are people in industry that are not on the committee, but they come together and they provide their input and expertise. So the committee then gathers that information and uses it when they write or revise standards. Now for this standard, USP 797, a expert panel was created. This is for pharmaceutical compounding, sterile preparations, and this panel, well, the expert committee started the review of USP 797 back in 2010, and then they created an expert panel. Actually, they had a previous expert panel that was looking at some of the revisions that might be possible, and they had passed that information on. Well, in 2013, they created a new expert panel with new people, and these people were across industry. There's people from pharmacy compounding accreditation boards. There's people from animal laboratories where they, because some of these compounded preparations are used in animals. So there was someone from there. There were people that worked in hospital pharmacies and did compounding. There's just a wide variety of people, even someone from the VA. And I happen to be on this committee as well. It was an application process, and they picked experts across industry. Now this expert panel that I was on that was started in 2013, we had meetings both online and in person, and we reviewed USP 797. And USP had received some comments through the years since it was first, well, not since the first time it was established, but the most recent version of USP 797 was published before 2010. And so there were people that were users specifically that were, and at USP they call them shareholders. They were reviewing, using, the standard and then making comments. There was also, the FDA has been involved, and in there, how they go out and do audits, and you've seen some of their findings, their audits that have been published as 483s and warning letters, and there's been some compounding pharmacies in the news. Well, all that information is taken and it was brought to the expert committee, and then the expert panel that was created also saw this information. Now, as the expert panel met, we went through recommendations. The expert panel also made recommendations as well, and we compiled all this information and then started filtering through it and pulling out what we felt was most important and providing additional information. And the expert panel ended up compiling about a 20-page document that was then passed on to the expert committee for their review, and they're going to consider the recommendations of the expert panel. As I mentioned before, there's a compounding expert committee, and they are the ones this expert panel was reporting to, but also involved is a microbiology expert committee. And so between the two of those expert committees, they will look at the comments the expert panel made and go off those recommendations to update USP 797. Now, I know at least the microbiology expert committee is meeting in, I believe it is towards the end of October, and I believe the compounding expert committee will be meeting basically at the same time, and they'll be reviewing these comments and considering what they will move ahead with in changing USP 797. Now they don't have a time frame set for that revision, so that's one piece of information I can't give you right now, but they will publish revisions and comments in the Pharmacopoeia Forum, there we go, the PF, and so then there'll be time for everyone else in the world to make comments on it. So that's all the background. Now I'll really go into some of the recommendations that were made by the expert panel, so you know what changes might be on the way. Now again, these are just recommendations, and the expert committee, will decide which recommendations they will accept and which ones they won't, and so on and so forth. We'll just go through the rest of these slides, are just is a summary of the recommendations that were made. So first of all, there were just some general comments made, and it was specifically about enforcement of USP 797. It was clarified to the panel that 797 only applies to non-outsourcing pharmacies. So if the pharmacy is outsourcing, then the standard doesn't exactly apply. We also asked that there be more enforcement of the standard, and we're told that the Drug Quality and Security Act is in place, and that gives the FDA the right to go into these compounding pharmacies, but they don't have as much ability to enforce 797 as they can on other rules because of the Security Act, and a lot of these, well, all of the states have compounding boards that go in and do inspections and things anyway, so the FDA relies on them, and they usually only come in case something big is happening. Now, we also suggested many changes to the definitions, specifically those related to the environment and classification, because some of them were very vague, and then others were didn't fall in line with what the other industries that use these call them. So some of the suggested definitions to be changed were clean rooms, anti-areas, buffer areas, and aseptic processing. The next area that was recommended, there was a lot of information that went into it, a lot of input, I should say, was to have a stronger requirements for what an expert is. USP 797 explains that an expert should do the training. Well, there wasn't exactly a definition as to what that is, so it was recommended that qualifications be put with that. Also with the personnel training and evaluation, we asked for an annual written examination and to use qualitative and/or quantitative analysis for the personnel evaluation. Now each one of these bullet points, the large bullet points, are sections within USP 797. If you're familiar with it, you'll recognize these. There are also recommendations made on single-dose and multi-dose containers. A couple of the more specific ones were to address commercially manufactured pharmacy bulk packages, and then also that single-dose containers not be used to prepare multiple syringes. As far as verification of the compounding accuracy and sterility, some of the recommendations were to specify storage times, to harmonize with the Microbiology Expert Committee's Depyrogenation chapter, which is USP 1228, because there's conflicts between the two. Also, there's a slight differentiation between aseptic processing and terminal sterilization, and we felt that language needed to be stronger and make a better separation between the two. And in this area, there were several items also that needed to be addressed specifically by the Microbiology Expert Committee, and one of the members of that committee was on this expert panel, and so he said that they would, actually I think there were two that were on this panel, and they said they would take it back to the expert committee and address those. So there's a lot of things that are depending on another committee to do before this standard can be changed. Now, as far as environmental control, there's not a lot of information in USP 797 about how to design and construct a classified area. So we suggested that that IEST and CETA be consulted, or to consult with other experts in the field that design and construct clean rooms for USP 797. We also suggested that there be a subchapter or some informational chapter further in USP that go over points to consider when designing and constructing a clean room. Also, there were a few specific suggestions made, like to specify HEPA filtered air has to be introduced at the ceiling and not from a mechanical room, because a few of the members of the panel said that they do inspections and they find that there is air coming from a mechanical room rather than as it should. So another one was just to make a clear distinction between the controlled environments and their function. Continuing on, there isn't a lot on leak testing for HEPAs, especially when that should be done, so requested that it be made after installation. There's a lot of things surrounding the validation certification of those controlled environments within a USP, well, within a compounding pharmacy that aren't addressed, and so we wanted to make sure that there's a clear distinction between how to validate and certify and classify and monitor. Also, there's emergency situations that happen that need to be discussed, that need to be in a company's standard operating procedure, so they know how to shut down and bring back up the clean room in case there's an emergency situation, for instance. And there were some things listed in USP 797 as to what you can take into the clean room, but there's no list of things that should not be allowed in the clean room, and so there was we recommended that be put into place as well. Still going on with the environmental quality and control section, we made specific recommendations and requirements for the viable and non-viable testing. We said it needed to be clarified and strengthened, specifically to describe the nature and frequency of testing, to make sure that right now for viable sampling it's very vague and almost leaves it that you could do it annually, and some people are only doing it annually. The suggestion was to do it at least monthly, if not more often, and that should all be based on risk assessment. We also asked that they address how to respond to positive results if limits are exceeded, what to do. That's not explained in USP 797, nor is trending very well. Just says you need to do it, so we suggested that more language be put in to clarify that. We also suggested that it be aligned with USP 1116, which is the aseptic processing clean room chapter that talks about sampling and gowning and all those things. So those are some of the things that we had recommended there. A few more. The section's a very large section, and this is the area that we hit hard. So had to make sure that that the cleaning requirements be clarified, including the sterile compounding area. Again, not very explicit at the time. More things on garbing, gloving, audits. It was suggested that the media fill test or the simulation test that's done be revised to be more in line with ISO 13408-1. rather than what's currently in there, which is not much. Another chapter that we've suggested is that one be written on clean room management for compounding. And then also last is to address environmental monitoring frequency based on the scale and number of personnel in the room. Here's just some other recommendations that we made. There's an area in the chapter that talks about standard operating procedures, but there's not. It talks more about what should be in a procedure and some of the elements. We suggested that required standard operating procedures be listed, specific ones, you know, in general, with general categories or headings, but be more specific than what's, excuse me, than what's already in the document. One of the things that people have seen is that people will use waterless hand scrubs and use it on the gloves, and so a point we wanted to clarify is that that's not appropriate. Also clarifying the types of wipes that are used for cleaning, and then just some other information such as additional guidance on sterility testing, how to address potency over time versus stability, and one thing that a few of them thought needed to be added was general guidelines on assigning beyond use dates. So again, all this information was given to the expert committee. Oops, excuse me. Oh, here's a couple more. Also better define endotoxin limits, especially for beyond use dates, and to update the common disinfection section to include newer disinfectants and sterilization methodologies. So again, all this is a brief, fast summary of 20 pages worth of information and recommendations that the expert panel came up with. Actually, there was more than 20 pages. We consolidated it down to 20 pages. And then this was given to the expert committee. Now the expert committee will look at it, as I said, with the compounding expert committee and the microbiology expert committee will work together to decide which recommendations they will use and which ones they won't, and then they'll come up with a revised USP 797, which they'll put out in the Pharmacopoeia forum, and then we'll have all of us that use it can then make comments on it, and they'll take those comments and incorporate them into the document further, or there may be more discussion time period and things like that. So that is a quick look at that entire document. Now there's a few other things that we want to bring up here. An additional standard to look out for is I'm involved, and there's several others at IST that are involved in a new working group that we have. It's Working Group 48. And the title of it is Guidance for Design, Performance, and Operations of Controlled Environments per USP 797. Now we're working on providing more information for those that use 797, more specifically to controlled environments. So designers, facility directors, and those directly involved in the pharmaceutical compounding area can have more information on controlled environments. And this working group will be meeting at the 2014 IST Fall Conference, which is being held in Cary, North Carolina at the end of October, last week of October. And you're welcome. If you're not a member of IST, please come and join us for that. Now, a lot of people know that IST is synonymous with clean rooms, but in case you don't, here's a little bit more information. And there's training courses that are coming up at the fall conference, and these are included. Again, you'll get these slides, so this information will come to you. So, any questions?